In this video, we're going to take the concepts that we learned from the last video, from Mendel's experiments and the concepts of alleles and representing them with letters, and we're going to make some predictions about offspring made from parents with certain genotypes. We're going to predict their phenotypes, we're going to predict their genotypes, and make some ratios. So we're going to start off with some simple monohybrid crosses, which basically means just comparing one character at a time. So we'll maybe predict height alone or eye color alone, and then later on when we get more confident, we'll try dihybrid crosses, which basically means comparing more than two characters, or two characters at once. So eye color and height at the same time. And you could even expand that, do trihybrid, tetrahybrid, as many characters that you might be dealing with. So monohybrid first for the majority, and then afterwards we'll go into dihybrid. You'll always see these slides at the beginning of a presentation just to help you, or just to refresh your memory about what characters are and what traits are. So make sure you review that. And remember, we're focusing on inherited traits, which are traits that can be passed on from generation to generation. We wouldn't be able to predict traits that are not heritable. So if you dye your hair, your offspring will not get that hair color because it's not part of your genotype, your genes. So when Mendel did his experiments, he realized that inheritance is really a probability. It's a likelihood of something happening. It's the chances of something happening. It's not necessarily a guarantee. So, for example, he noticed that, let's say you had an organism with genotype big T, little t, half of your offspring would, half of your gametes would be big T, and the other half will be little t. So, roughly 50% of the time you should be transmitting big T, and roughly 50% of the time you should be transmitting little t. Now again, it's a probability, it's not exactly right on, but it'll be about 50-50. The more offspring you have, the closer you'll be to this probability. It's kind of like flipping a coin. If you flip a coin, let's say 2 times, it's possible to get heads twice. It doesn't mean your coin's a trick coin. But the more you flip that coin, the closer you'll get to the 50-50 chance. So for example, if you flip it 100 times, you might get 51-49. You might get 48-52. Closer and closer as you have a larger sample size. Now the idea is with this is that if you have a 50% chance of getting big T and a 50% chance of getting little t, you can make predictions about what your offspring might end up being. So let's say you had 2 individuals that were big T, little t, cross them together, and you want to figure out the chances of getting big T kids. Well, you have a 50% chance here and a 50% chance here. So usually when you want to figure out what are the chances of getting 2 things to happen together, this and this, you would multiply them. So you would do 0.5, 50% chance, times 50% chance, and that would give you a 0.25 chance, a 25% chance, or a quarter chance, 25%. And so you can see that in this picture over here. You have a 50% chance of getting this, 50% chance of getting this, combine them together, and that would be a 25% chance. You can actually do that with something called a Punnett square. A Punnett square is a diagram that you can use to predict the expected proportions of offspring based on the parent genotype. The way a Punnett square works is you put the gametes of the parents on the top, so let's say this is from the mother, and also on the side, let's say this is from the father. Half a chance of getting big T, half a chance of getting little t, half a chance of getting big T, half a chance of getting little t, and in the boxes that you see here, you have the fertilization event. So if this gamete is given with this, you get big T, a tall plant. If this gamete is given with this gamete, you get big T, little t. If this gamete is given with this gamete, you get big T, little t. So there's actually 2 ways to get big T, little t. You can get little t from the egg and big T from the pollen, or you can get big T from the egg and little t from the pollen. That's why there's more heterozygous produced than there are homozygous, because there's 2 ways to get the heterozygous individual. And then let's say you get this gamete with this gamete, then there's little t, little t. and you multiply the chances together to get the chances of each offspring. Or you could also just look at it visually. You have one of these big T out of 4 boxes. For the big T, you have 2 out of 4 boxes, so 50% chance of getting heterozygous. And then for the little t, you have 1 out of 4 boxes. Now, if you were to look at the 4 boxes here, how many are tall? Well, you have 3 tall ones to 1 short one. And so what we're going to do in this lesson is we're going to learn how to use these Punnett squares and these probabilities to figure out what type of babies you can make and in what proportions you can expect them. Again, it's not a guarantee. You might get 75% tall and 25% short, but it might not be exactly 75%. It'll be pretty close, but not exactly, because again, it's all a probability as opposed to a guarantee. So before you can do Punnett squares, you need to be able to understand genotypes and phenotypes, how they relate together, and we did many problems like this in the previous PowerPoint, but we'll just do one quick little refresher slide here. You can pause the video and try it yourself. So here we have the scenario, big B brown is dominant to little b blue. If you have big B, little b, what's the individual's phenotype? Well, you should know that it's brown because brown is dominant to blue. If you have big B, same idea, because there's only some brown alleles in there. And if you have little b, well, that's the only way that the blue recessive trait can be shown, so it would be blue in that case since there's nothing masking it. Here we can see a white tiger. Its phenotype is white. Its genotype is little o because the white allele is recessive. If it had a dominant allele, then you know your tiger would be orange. That could be either big O, or big O, little o, heterozygous. But either way, dominant would make it orange. So you can see here, we have an orange tiger, and it would be big O, little o, or big O, big O. Over here we have some P's. Yellow is recessive, so it would have to be little g, little g. To have green, you need to have big G, little g, or big G, big G. Albinism is a condition where individuals cannot make the appropriate amount of pigmentation to get darker skin, so they lack the pigmentation. They don't have the genes required to do that. Albinism is actually a recessive trait. People who have the albinism phenotype, they don't make enough pigmentation, they don't make enough melanin, so they're more likely to get sunburn and sun damage on their skin, so they have to take extra precautions. But let's say someone did have albinism, you should be able to come up with their genotype, which is little a, since it's recessive based on the information given here. If someone did not have albinism, then you'd know that they either be big A, or big A, little a. Now, when I say having albinism, this is different than just being Caucasian or a different color. This is you actually lack pigmentation in your skin, but also in your hair and other areas as well, and you're more likely to get that sunburn. Again, from the previous presentation, remember that just because something is a dominant allele, it doesn't mean it's the more common one or the better or the more powerful one. It just means it's the one that's more expressed. So for example, here we have a condition called dwarfism. And believe it or not, dwarfism is actually caused by a dominant allele, the big D allele. So if there's someone who's a dwarf, that person is big D, little d, or big D, big D. If you're not a dwarf, then that's right, you guessed it, your genotype would be little d, little d. So again, dominant does not mean more common, does not mean necessarily better or more powerful, it just means it's the one that's expressed. You also need to know how to figure out what type of gametes can be produced by individuals, which we saw in the previous presentation. If someone is homozygous, big G, well, all they can make are big G gametes. That's it. Remember, the gamete is represented by one letter because it's haploid, whereas the individual is represented by 2 letters because it's diploid. And you can see that the homologous chromosomes separate, but since they had the same alleles, they all make 100% G gametes. So this individual can only pass on big G. If the individual was a heterozygous individual, then it could make either big G or little g, 50% of each. You can see the law of segregation here. The homologous chromosomes separate, so the alleles have separated. The big G and little g have gone in separate cells. So alleles of a given gene will always separate. They'll never be inherited together. So you should be able to try an example like this out. So determine the gametes produced by the organism with the following genotypes. So let's just select one sample problem. I'll do big M, little m. So if I was big M, little m, that means one of my chromosomes has big M on it. Remember, DNA duplicates, that's why there's two big M's there. And the other one has little m on it. Keep following that through. The blue one went in one cell, big M. The yellow one went in the other cell, little m. And then the sister chromatids separate, so we have big M over here and little m over here. So that means we have the gametes that are possible, big M or little m, for the heterozygous individual. And you can do that for any one of these genotypes here to figure out what type of gametes are produced. But in reality, when you're doing a problem, you won't need to draw this out. You'll just have to tell me that big T, little t can make big T or little t. I will ask you eventually to be able to relate it to meiosis, but for quick problems, you can just remember how that works. So if you take a look here, and I asked you what type of gametes individuals can make, you should know that this makes big B or little b. You should know this individual can make only, that's right, big B, and this individual here can make only, yes, little b. So these are the general rules for making a Punnett square. It's a 2 by 2 table. So there's 2 by 2, which means there's 4 squares within it. Gametes of the parents go on top and on the side. So you might have father on the top, mother on the side. Doesn't really matter which one goes where. You'll still get the same answer in the end. And inside the boxes, you have the fertilization events that occur. So we'll do a few problems together, but the idea is that if you had a big B, little b cross a big B, little b, to find the kids, you would say, well, if big B goes with big B, well, in this box I'll have big B, big B. If little b goes with big B, then in this box I'll have big B, little b. If little b goes with little b, then in this box I'll have little b, little b. And then this big B goes with this little b, then in this box I'll have big B, little b, the heterozygous individual. 2 heterozygous here, 1 homozygous dominant, 1 homozygous recessive. So you simply put the genotypes, the gametes on the top and side, and then you match the fertilization events to see what are the probability of getting the different type of offsprings. Now, I might ask you for the genotypic proportions and the phenotypic proportions, or the ratios as well. Proportion just means the percentage of each genotype if I ask you for genotypic proportions. And for phenotypic proportions, it means the percentage of each phenotype. I might just ask you for the ratio. The ratio would simply mean how many to how many. So it might be, for example, if I asked you for the phenotypic ratio, I might say 3 brown, that's the phenotypic ratio, to 1 blue, little b there. That's phenotype though, not genotype. If I were to ask you for the proportion, you might say 75% brown, you'd have to write down brown, and 25% chance blue. So if there were 100 individuals in the population, 75% of those 100 individuals, so 75 individuals would be brown eyes, and 25% would be blue eyes. I could also ask you for genotypic ratios. So a genotypic ratio, for example, if we're looking at this particular Punnett square here, is the ratio of genotype. So one big B for every 2 big B little b for every 1 little b little b. I could also ask you for the phenotypic percentages. So if the genotypic percentages, so 25%, you'd have to write down big B, little b. 50%, sorry, you'd have to write down big B. 50% big B, little b, so you'd have to write that down. And then 25% little b, and you write down little b after the percentage. So those are genotypic percentages or proportions. These are genotypic ratios over here. 1 big B for every 2 big B little b for every 1 little b little b. And again, it's not a guarantee, it's simply a probability. So we're going to take a look at a Punnett square to see Mendel's experiment. So he started off with pure breeding plants. So let's say they were pure breeding tall plant, big T, crossed with little t. So let's put the gametes of the tall one on top. So big T, and let's put the gamete of the short one on the side, little t. Now what we're going to do is we're going to combine the big T with little t, so this big T here with this little t here, big T, little t, this big T here with this little t here, big T, little t, this big T with this little t, so big T, little t, and this big T with this little t, so again, big T, little t. So if I were to ask you for the genotypic ratio, it's really difficult to make a ratio here because it's only one type. So you could say all big T, little t. The frequency is a bit easier, or the proportions are a bit easier. You could say 100% big T, little t. For the phenotypic ratio, again, you don't really have a ratio because they're all of the same type, but we'll say all tall. For the phenotypic frequency, or the percentage, you can just say 100% tall. Remember that tall was the dominant allele in this experiment. Notice genotype versus phenotype. And again, these were our F1 generation. So now we'll do the second experiment where Mendel had crossed the F1 individuals together, which was big T, little t, crossed with big T, little t. So I'll put the one parent on the top here, big T, little t, its gametes. The other parent on the top, on the side here, big T, little t, its gametes, and do the crossing. So big T, big T, big T, little t, big T, little t again, and little t, little t. So we can start off with the genotypic ratio. And what's interesting is whenever you see this genotypic ratio, you know that it must have been heterozygous individuals crossed with each other. So we get one big T, so it's always one homozygous dominant, for every 2 big T, little t, for every 1 little t, little t. And so if you want to write that in a frequency or a proportion, you would just write 25%, make sure you put big T, 50% big T, little t, and then 25% little t, little t. That's for the genotypes. For the phenotypes, it's a bit different. You need to look at the ratio carefully. So remember that big T is tall and it's dominant. So in this Punnett square, you have 3 tall individuals. So 3 tall and only 1 short for every 1 short individual. And so if you were to write that as a percentage, then that would be 75% tall. So you'd write tall next to that, 75%, and 25% short. And so that's our phenotypic frequency or phenotypic proportion. Whenever you cross heterozygous individuals, like you see here, your phenotypic ratio, your Mendelian phenotypic ratio is going to be 3 of the dominant phenotype to 1 of the recessive phenotype. And then for the genotypic Mendelian ratio, it's 1 big A, 2 big A, 1 little a, little a. Now for the phenotypic ratio and the Mendelian ratio, another way to sometimes write that is 3 big letter, empty space, to 1 little letter, little letter. I know this is written in the genotype form, but it still represents the phenotype. And here's why. The 1 little t is a recessive phenotype. The 3 big T with an empty space there is actually the dominant phenotype, because this big T could be either the big T, because there's 2 of those, or the big T, because there's 1 of those. And when you add them together, that gives you your 3 individuals with the dominant phenotype. So it's important that you remember that 3-to-1 ratio, because whenever you spot it, you know that the parents were heterozygous individuals. So you don't have to do as much problem solving if you know that the parents were heterozygous individuals to begin with. Sometimes I'll ask you to predict what the kids will be, but sometimes I might give you the kid ratio, the offspring ratio, and you have to figure out what the parents were. So we'll do an example together here. A geneticist crosses 2 pea plants. One of the plants is heterozygous for the dominant inflated pea pod trait, and the other has the constricted pea pods. So in this question, I know inflated pea pod is dominant, and constricted must be recessive, because it told me that the other one is dominant. So I'll give that allele A. What would be the expected genotypic and phenotypic proportions of the offspring? So first of all, you have to figure out what the parents are. We told you that one of the parents is heterozygous. So the genotypes of one parent is big A, little a, and the other parent is going to be constricted pea pods, and we know that's recessive, so little a, little a. And so I always write down a summary of what the cross is going to be. Here we're going to have big A, little a, crossed with little a, little a. And we're going to make a Punnett square to see what the offspring could be. But we need to figure out what our gametes are. So one parent can make big A, little a, that's this heterozygous individual here. And so I'm just doing this work on the Punnett square. The other parent can only make little a, little a. So we'll put little a here and little a here. And then we start the fertilization event. So we have big A, little a in this box, big A, little a in this box, little a in this box, little a in this box. And we can start off by writing out our genotypic ratio. We're going to have 2 big A, little a for every 2 little a, little a. And you can simplify that. It's a 1-to-1 ratio. It's a 1 big A, little a to 1 little a ratio. Basically, that means 50-50, right? Half our big A, little a, half our little a, little a. If you were to write that down in the genotypic frequency or proportion form, you'd have 50% big A, little a. Make sure you write that down. And 50% little a, little a. You can also do a phenotypic ratio. So A is inflated, so we have 2 inflated for every 2 constricted, and you can write down 1 to simplify it, inflated, and yes, you will need to simplify it, for every 1 constricted. And again, you can write that down as a proportion or frequency, so we're going to have 50% of them that are inflated, and 50% that are constricted. So here's another ratio you can remember. It's not quite called the Mendelian ratio. It's not the 3-to-1 ratio, but it is a 1-to-1 ratio, a 50% chance of getting both phenotypes. If you see that both phenotypes occur in a 1-to-1 ratio, 50-50 chance of all phenotypes, then that means that one of your parents was heterozygous and the other was homozygous recessive. So spotting these patterns will make your problem solving a lot easier on quizzes or tests or assessments. So this is another sample problem. In humans, the trait for detached earlobes, big D, is dominant to the trait for attached earlobes, little d. An individual with attached earlobes is crossed with an individual homozygous for detached earlobes. So you can try a Punnett square to figure out what the proportions of offsprings are for this cross. So first of all, figure out what the genotypes of your parents are. We have little d for attached and big D for detached, because we're telling you that the person that's homozygous for detached earlobes, the person with detached earlobes is homozygous for it, so you know it's not big D, you know it's big D. So it's about reading those questions carefully. The individual with attached earlobes, I knew its genotype because they told me it was recessive, and there's only one possible recessive genotype, so that's how I knew the genotype for that parent. The possible gametes for each parent is what you need to figure out next. So little d can only make little d, and big D can only make big D. So we'll just take a look at what that looks like at a meiosis level. Here we have the individual that's, let's say this was a heterozygous individual. That heterozygous individual could produce either big D or little d. Now we don't have a heterozygous individual. We have two homozygous individuals, and that's why they can only produce one of each. So another question I could ask you in that scenario is, what are the phenotypes of the parents? Well, based on the genotypes, I told you that there's someone with attached earlobes, little d, that's a phenotype, and big D is detached earlobes. So there it's just simply a test of your knowledge of the term phenotype. And then I could ask you, what will the offspring be when you do this cross? So you can do a Punnett square. You can put one parent on the top, big D, big D, one parent on the side, little d, and then combine all the gametes together, and you get your 100% big D, little d. So for the genotype frequency or proportion, we have 100% big D, little d. This is like a pure breeding example that we saw with Mendel. So it's 100% heterozygous. Our genotype ratio, well, it's all big D, little d. Our phenotypic ratio, well, it's all detached, or 100% detached if it's proportion form. So the idea behind crossing means this. So when crossing simply means mating, so when the big D mates with the little d, they each contribute the only gamete they can make, big D and little d, and you have a fertilization event. So big D sperm will fertilize little d egg, or the other way around if it was the father that was little d and the mother that was big D. And after fertilization, you have your zygote with big D, little d at the end. And that individual, through mitosis, will be an individual with detached earlobes. So there are some more sample problems that you can attempt in this PowerPoint. I'm going to do this one with you. You can pause it, try it yourself first, and then take a look at your answers and compare them to mine. So we have dwarfism, which is dominant in humans, and you're told that there's a heterozygous individual crossed with a normal individual, not a dwarf. And so there's a lot of information given in that statement. The heterozygous individual, so what are the genotypes of the individuals in the cross? Well, let's just say we have big D, little d for the heterozygous individual. The other individual is not a dwarf. We know dwarfism is dominant, so if you're not a dwarf, you have the recessive alleles. So the heterozygous individual, they're asking for the phenotypes, big D, little d. That individual, since there's a dominant allele, we know that individual is a dwarf. And then the normal individual, well, that's the little d, not dwarf. What are the genotypic and phenotypic ratios? So if we cross these individuals, what will their genotypic and phenotypic ratios be? Then we're asking you, what are the chances of having a dwarf child? what are the chances that they have a normal child? And if they have 8 children, how many would you expect to be normal? How many would you expect to be dwarfs? So we'll do a Punnett square to solve this problem. So we have big D little d with little d little d. Now already you can probably predict what the answer would be based on the fact that we did a sample problem like this before. Remember when you cross heterozygous with homozygous recessive, you get a 50-50 phenotypic ratio. You get 50% would be dwarf and 50% would be normal, or sorry, I should say 1-to-1. One dwarf for every one non-dwarf individual. And the genotypic ratio, again, is also 50-50. You'll get one, let's try this out, so you get your big D little d and your little d little d. So one big D little d for every one little d little d. So we'll actually prove that in a Punnett square. So we'll put the big D little d parent on the top here. We'll put the little d parent on the side here. And we'll do the fertilization event, big D little d, big D little d, little d little d, little d little d. So we have one dwarf or two dwarf for every two normal, which is 1-to-1. So if we were doing our phenotypic ratio, we have one dwarf for every one normal or non-dwarf. And then genotypic ratio, we have one big D little d for every one little d little d. What are the chances of having a dwarf child? Well, you look at your Punnett square, dwarf is big D little d, and there's 4 squares, so that's 2 out of 4. 2 out of 4 chances, or you can say half a chance, or 50% chance. A non-dwarf child, well, there's again little d twice out of 4 boxes. So it's the same idea, 2 out of 4 chances, or 1/2, or 50%. So you could extend this to answer the next question, which is, if they have 8 children, how many will be dwarf children, and how many will be normal, non-dwarf children? So you would have, if you have 50% chance of getting non-dwarf, that would be 4 normal, I'll just put 4n, and 4 dwarf, because it's a 50-50 chance. So I just multiply the percentage of each phenotype by the total number of children that are given in the question. So there's a few more problems that you have over here. These are from your textbook, so you can go and do the tutorials to see the answers for these and carry it out to make sure you understand the concept. You need to be able to do a Punnett square. I know some of you might be able to do this in your head, but it's always good to be able to show your work to show how it's happening. I'll do this problem with you. First you can pause the video, try it yourself, and then you can take a look at my answers. So we have a pea plant, the allele for yellow seed color, Y is dominant over that of the green color, little y. Consider a cross between a pea plant that is homozygous for yellow, so big Y, and a plant that is homozygous for green seeds, little y, little y. Sometimes your uppercase letters and lowercase letters might look similar. Make sure you really differentiate between them. For example, it's sometimes hard to tell between uppercase O and lowercase o and uppercase e and lowercase e, so make sure you differentiate between them. Where possible, try not to select letters that are too similar in their uppercase and lowercase forms. Try to choose things that are a bit more different, like E or maybe D or A, so on and so forth. So create a Punnett square to determine the possible genotypes and phenotypes of the offspring. This one's a pretty straightforward one. We'll put our gametes on top here for one of the parents, and then our gametes on the side for the other parent. Little y, then you can combine them together. So big Y with little y, 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 and you can predict that we have a 100% chance of having the yellow peas. Again, some more problems here. So we have 2 heterozygous yellow pea plants this time. and you want to figure out what their genotype and phenotype ratios are in the next generation. So in the F2 generation. Our F1 generation, that's our parents that we're dealing with. F2 are the offspring. And so already you should probably be able to guess what it's going to be. You should know this is a case of the 3-to-1 ratio. We should have 3 of our dominants, 3 yellow, for every 1 green. And I know that because there are 2 heterozygous individuals being crossed. If I want to do my phenotypic ratio, I'd know there's 1 dominant genotype for every 2 heterozygous genotype for every 1 homozygous recessive phenotype. These are my Mendelian ratios for heterozygous crosses. You could also do your Punnett square to prove that that's the case. So big Y, little y on top. Big Y, little y on the side. So we have big Y here. If the top gives the big Y and the side gives big Y, we have big Y, little y here. If the top gives a big Y and the side gives a little y, we have big Y, little y over here if the side gives a big Y and the top gives a little y. So 2 ways to get the heterozygous individual, depending on which parent gives which allele. And then we have little y over here if the little y gives if the little y is given by the top and by the side as well. And so just as predicted from our knowledge of the Mendelian ratio with heterozygous individuals, we get these ratios that we predicted up here. So you should be able to do problems like this now and make the predictions. This is, again, Mendel's experiment with the different plants. We already did it before, but the idea is that the parents that we were crossing were pure breeding. One was tall for the phenotype, one was short for the phenotype, and they were pure breeding. So what that means is the tall individual had big T, and the short individual had little t. If you do a Punnett square, then you know that your short phenotype will disappear, and it's because all the plants are inheriting big T from the pure breeding tall. They're also inheriting little t, but the big T is dominant. And again, you can figure out your phenotypic ratios and your genotypic ratios and percentages. So our genotypic ratio, well, it's just all big T, little t. It doesn't make sense to write a ratio. I mean, you could do for big T, little t for every 0, everything else for a genotypic ratio, but it doesn't really make sense to do that right now. Genotypic percentage, then you can just put 100% big T, little t. And then F1 phenotypic percentage, 100% tall. And we know that the F1 offspring are heterozygous. They contain 2 different alleles, big T and little t. You then take the F1 and you cross them together. So we have tall phenotype crossed with tall, but they're not pure breeding this time. They're hybrids. They're heterozygous. We have big T, little t crossed with big T, little t. And we can figure out what the offspring will be. We can put big T up here, little t up here for one of the parents, big T over here, little t over here for the other parent. And then you simply do the fertilization in each box. So big T, big T, big T, little t, big T, little t over here, little t, little t. The F2 genotypic ratio, it's overlapped here, but let's just take a look. For the genotypic ratio, we'd have 1 big T for every 2 big T, little t, for every 1 little t. You could do your percentage. You have 1 out of 4 big T, so it's 25% big T. You have 2 out of 4 that are big T, little t, so 50% big T, little t. Then you have 1 out of 4 that's little t, so 25% little t. So in your population, if you had 100 individuals, 25 of them would be little t, roughly. The phenotypic ratio for the F2 generation, so we have 3 tall for every 1 short. 3 tall, which is our dominant trait, for every 1 short. And then our phenotypic percentage at 75% tall and 25% short. And you can see the 75% tall come from the genotypes with the dominant alleles in them. You could also write down the phenotypic ratio as 3 big T with a space there to show that could be little t or big T in there. And then 1 little t. I know it looks like genotypes again, but it represents the dominant phenotype for the recessive phenotype. And that's our Mendelian phenotypic ratio. So this is super important to remember, this phenotypic ratio when you cross heterozygous individuals. 3 dominant traits for every 1 recessive trait. And your genotypic ratio is your 1, for example, big A, homozygous dominant, for 2 big A, heterozygous, for 1 little a. So make sure you remember these ratios for the heterozygous individuals. It will really save you a lot of time when you're doing some problem solving to try to figure out what the parents are. Remember that the ratio applies to more than just 3 to 1 for individuals total, but to cases like this. If you have 30 to 10, that's 3 to 1. That means you know the parents were heterozygous. 27 to 9, that's 3 to 1. That means you know the parents were heterozygous as well. If you get this genotypic ratio, and I didn't tell you what the parents were, you know they must have been heterozygous, because that's the ratio that you get for genotypes when your parents had the genotype, had the heterozygous genotype. So we'll do a sample problem here to apply that logic. In aliens, green skin and blue skin are possible, and across, 89 green aliens are produced and 30 blue aliens are produced. Use a Punnett square to justify your answers to the questions below. What are the most probable genotypes and phenotypes of the parents? So take a look carefully. We have 89 green aliens for every 30 blue aliens. If you look carefully, that looks like 90 to 30, which is 3 green to 1 blue. This is a 3 to 1 phenotypic ratio. That means that chances are we had heterozygous parents. That means our genotype, so let's just give a letter to green, and blue. That means our genotypes were probably big G, little g for one and big G, little g for the other. Now for phenotypes, we need to figure out which one was dominant. And I kind of already gave that away when I gave the letter green, big G. How did I know it was dominant? Well, remember that in the 3 to 1 ratio, the one that has 3 is the dominant one, and the one that has 1 is the recessive one. So green must be the dominant phenotype, the dominant allele. So that means if my parents are heterozygous, that they have a dominant allele, which means that they must also be green. So you can use ratios to support your answers to what the parents would most likely be. And so in that explanation, I explained which was dominant, green, and which was recessive. So when you're answering a question on a test, you need to make sure you clearly tell me green is dominant, and then tell me blue is recessive. So answer both parts of the question. Don't lead me to assume that I'm going to think that the next one's recessive just because the first one's dominant. Maybe you don't know what these words mean. Maybe you wanted to say that they're both dominant. And you don't know what you're talking about. So I need to make sure that you're clear with everything. And then you can give the explanation with the 3 to 1 ratio. What are the genotypes of the offspring produced in the question? Well, you would know this too. There's 89 green for every 30 blue. You know that of the offspring produced in the question, you would have 1 big G, 2 big G, little g, and 1 little g, little g. So that means about 25%. Let me get a different color here. 25% would be big G, big G. 50% would be big G, little g. And 25% would be little g, little g. So you have about 100 individuals. I'm just rounding up. That means 25 of them would be big G, big G. Now, if you want to find the exact number, you'd simply do 25% times 90 times, sorry, you don't have 100 individuals. You have, let me just take a look here, 120 individuals, I should say, because you have 90 plus 30, 120 individuals. Right, so 89 green plus 30 blue, that's 120 individuals. And you need 25% of 120, which would be about 30. 30 are big G, big G. 50% of 120 would be 60 big G, little g. And then 25% again, 30 little g, little g. So when you add those up, you get 30 plus 60 plus 30, that's 120. So you can figure out how many of those individuals in there would be each genotype. So in our 89 green, 30 are big G, and 60 would be big G, little g, roughly. Because it's one big G for every two big G, little g, and that would make sense. And then of course you have 30 little g, because there are 30 blue. And that you can tell just from that phenotypic ratio. Here we have another example. In aliens, green skin and blue skin are possible, and across 1,000 green aliens are produced and no blue ones are produced. Use a Punnett square to justify your answer. What are the most probable genotypes and phenotypes of the parents? So this depends on several factors. Let's say I clarified the question and I told you that you had 1,000 green aliens that were made and no blue ones made. And I said that you don't have any other information other than the fact that maybe these aliens might have kids, maybe they won't. So let's just say for now they didn't have any kids. There's a few ways you can answer this question. Based on the previous question, we said that green was dominant. Green was dominant. Now, if we're continuing with that logic, we're saying green is dominant, and you say the parents are big G, little g, and big G, little g, then you would be wrong. And that's because if you do this, you know that you're going to get some blue kids. So you might say maybe the kids are big G, little g crossed with little g, little g. If you do this, then you would also be wrong, because you can also get blue kids. If you do your Punnett square, you'll get about 50% that are blue. So this would be wrong. You could say you had big G crossed with little g, little g. If you do that and you do a Punnett square, all will be big G, little g. So that's one possibility. You could also say you had big G crossed with big G, because they'll all be big G if you do your Punnett square. So the idea is that you can try to take a look at what the different possibilities are for crosses and see if your possibility makes sense by checking it with a Punnett square. In this case, I've already crossed out these two possibilities because I know I'm going to get some blue in there. These are the only two possibilities that will give me only green. So which trait was dominant from the previous question? Well, we know it was green. Which was recessive? We know it was blue. What are the genotypes of the offspring produced in the question? Well, we know in this case that it could either be this or this, depending on what the scenario is that we have. I'd have to give you more information, but I want you to think that there are multiple answers to some of the questions. Sometimes there'll be one definite answer based on the question. Other times you won't know all the information, and that's sometimes the case in genetics. When you're a genetic counselor, when you're trying to figure out conditions that might be passed on from generation to generation, or whether something is dominant or recessive, you might only have limited information based on the current generation that you're working with, or the previous generation, and you need to see more information from the kids that are produced. So I want you to think about what you would need to know if this was the correct situation or if this was the correct situation. What information could confirm this as opposed to this one here? Think about that, discuss it with each other, do some research. That way you'll know, you'll really understand the concept. So this is our summary here. We're dealing with one trait, so we call it monohybrid crosses. If you deal with multiple characters, I should say, not traits, characters, then you'll be dealing with dihybrid crosses. Remember your Mendelian ratios. That'll help you answer questions that we saw in the previous slides much more efficiently than always having to do Punnett squares. Here are some more sample problems you can attempt. The answers are included in the space below. And then you have a few more practice problems that you can do on these slides with the answers on the next page. Now, one neat case that could arise is you need to figure out what your genotype is for something based on its phenotype. So here we have green peas. And if you have green peas, you know that you have a dominant allele, but you might not know what the next allele is. It could be a big G, little g, or it could be a big G as well. They could be pure breeding. And that's an important concept, because sometimes when you're buying plants or when you're buying animals, you want pure breeding. You want homozygous so that they weren't mixed with other types of individuals. And so you need to do a test to figure out, well, is my plant pure breeding, homozygous dominant, or hybrid, heterozygous? And so to figure that out, you could actually do something called a test cross. Now, you don't need to do that if the peas are yellow. Why not? Yellow's recessive. What does that mean? Yes, that's right. You need both recessive alleles to be recessive, to have the recessive phenotype. So there's no question. It's just little g, little g. But here the question is, which genotype are we? You can do a little procedure called a test cross to figure out which genotype you are specifically. And so we'll see what a test cross is in order to confirm which genotype we are. So a test cross is when you take an individual with a dominant phenotype. We don't know which genotype though. We know it could be big P, big P, little p, or big P, big P. And you cross it with a recessive phenotype individual, which has to be the little one, little p, for example. And the results will let you know if the genotype of the individual is, let's say, big P, little p, or big P, big P. It will confirm one or the other for you to see which one's pure breeding. So here are the results that you can get from this test. So we have a big P something, unknown. We want to know if it's pure breeding, purple. We cross it with a recessive. So if you get from this cross all 100% of the dominant phenotype, then you know that your plant was pure breeding because all the offspring were given the dominant allele. There was no recessive allele given. Okay, and it's not just a sample size of 4. You have a large sample size. That's why we can be certain of that. Because some students might ask, well, maybe the 5th one might have been white. True. That's why we do a large sample size instead of just a sample size of 4. If, again, in your large sample size, the other result happens where you get some purple, yes, but some white, that means that your plant was big P, little p. And over here, big P. Because that means that the purple flower was able to give some little p alleles to make little p with the white flower. So this would confirm that our plant is not pure breeding, but is heterozygous, and this would confirm that our plant is pure breeding, is homozygous dominant. So the test cross is always the dominant phenotype with the recessive phenotype, because the recessive phenotype has our known genotype, and the dominant has our unknown genotype. We don't know what it's going to be. And we actually still use this technique to figure out genotypes of organisms today, especially as it pertains to plants, but also other organisms too. And so here is the explanation again, just dealing with the idea that if only the dominant is made, 100% purple, 100% big P little p, and that means that this was our pure breeding individual. So here we have a sample problem that goes along with that concept. An alien green skin is dominant to blue skin. An alien has green skin. What are the possible genotypes for the green-skinned alien? Well, we have big G, little g, or big G, because it's dominant. If it's blue skin, only one possibility, little g, little g. Explain why there's more than one possible genotype for the green-skinned alien. Well, you should be able to relate that to the concept of dominant and recessive. The green-skinned alien can arise if you have both green alleles, but you can also have a green-skinned alien if you have a green allele and a blue allele, because the green allele will be expressed over the blue allele since the blue allele is recessive. Explain why there's only one possible genotype for the blue-skinned alien. Similar explanation. To be expressed, the blue-skinned alien must be paired with another recessive allele, otherwise the green dominant allele will be expressed instead. Describe a test you could use to determine with certainty which genotype the green-skinned alien is. What type of result would support the conclusion that the alien is big G, little g? What type of result would support the conclusion that the alien is big G, big G? Illustrate your answer with a Punnett square. So I'll do one of the Punnett squares with you, and you can do the other on your own. So let's choose our black color here. So if I did this, big G, question mark, crossed with little g, this is called a test cross. A test cross is used to help me figure out what the genotype of my unknown dominant phenotype is. And so if my individual was heterozygous, big G, little g, then what that means is that when I do the test cross, I get big G, little g here, big G, little g here, little g here. 50% of my aliens produced would be blue, and 50% would be green. Basically, the idea is that as long as I produce some blue, then I must have had the little g allele. If I didn't produce any blue with a large sample size, then I didn't have little g allele. I only had the big G. I only made green. But even if I don't see that 50% ratio, even if I just see one blue come out or two blues come out, then chances are that my individual, my alien, was heterozygous, because that's the only way you can make green. You need to have that little g there. To make blue, you need to have a little g there to combine with another little g over here. If you don't have that and you get 100%, let me just make that a clear Punnett square there. Let's erase this. And you get 100% green all the time, then that means you didn't have the little g to pass along. To the next generation. And you were a pure breeding green alien. Here are some more practice problems that you can attempt based on what we learned. Read the questions carefully, look at all the clues given to you. You always have enough information to answer the question. In the case that you don't, you should always kind of take the approach that you explain both cases. So if one scenario, explain what happens. If the other scenario, explain what happens. Here we have some more sample problems, along with the answers given to you on the side. So please try those out. Compare answers with your peers as well to make sure you're on the right track. Make sure you follow all the proper steps, show all your work. Don't just do it in your head. You need to make sure that you communicate what you're doing. Some more sample problems over here. A lot of these are in your textbook. You can always see the answers at the back of the textbook and then compare with each other to make sure you're on the right track with the explanation. And then a few other sample problems that are included here in the PowerPoint. So go over these problems, compare with each other. I'm going to try to go over this one with you. Try it first by yourself to make sure you understand the concept, and then come back and compare your answers. So in humans, tongue rolling is dominant. To non-tongue rolling, little r. 2 parents have 4 kids. 3 of the kids can roll their tongue, 1 cannot. write the phenotypic ratio of the kids. So we have 3 rollers to 1 non-roller. Based on your phenotypic ratio above, what are the most likely genotypes of the parents? Well, you have our 3-to-1 ratio here. So that means that they were both rollers. Mom was a roller, dad was a roller. And their genotypes would be they were heterozygous rollers, because that's the only thing that results in that 3-to-1 ratio. Explain how you determine this. Well, we know that it's because of that 3-to-1 ratio, the Mendelian ratio, and you can support your answer with a Punnett square. Here we have some more practice problems that you can attempt. Again, compare with your friends, your peers, to make sure that you're on the right track. So you have lots of practice problems. There'll be more posted online. The more you do, the better you're going to become at this. Initially, it might take you a bit of time to solve them and to make sense of them, but eventually you'll see a problem like this and be able to get through it within a matter of a minute or two. Here we have the case of dwarfism in humans with a sample problem that follows on the next slide. You need to be able to think a bit, know the terms, use the hints in the question, make Punnett squares, justify your answers, make sketches, and think carefully about what's being asked in the question. You've seen polydactyly before. Now we're going to apply a Punnett square question to it. Again, look at all the questions carefully, look at all the hints carefully, follow the information given within. The readings associated with these concepts can be found in pages 188 to 193. There are practice problems on page 193, number 1 to 10, that you should attempt, and there are also tutorials within the book that have associated sample problems. The tutorials walk you through how to do the sample problems, just like we did in this PowerPoint, so you may as well read through those. It's not additional work. Whatever you saw in this PowerPoint is in your book, so it's just a different perspective, and will give you more of an idea as to how to solve these problems. You should work on the practice problems in the PowerPoint, do the practice slide questions that I've included in the PowerPoint, and then additional worksheets will be posted online. The more that you do, the better you'll become, the more comfortable you'll be with answering these style of questions on the tests, quizzes, and assessments.